So in this video, I'm going to talk about DNA. So first, let's start off with the structure. So DNA is made up of building blocks called nucleotides. And there are 5 types of nucleotides. 4 are listed here: adenine, thymine, guanine, and cytosine. One not listed here because it's not in DNA is uracil, and that's in RNA, but we won't talk about that, so I'll just erase that. So we're going to talk about adenine, thymine, guanine, and cytosine. So purine bases, purines are adenine, which I'm going to abbreviate as A, and guanine, which I'm going to abbreviate as G. Pyrimidines are the other type, and that's the rest. So it's cytosine as a C, and then thymine. And a good way to remember that is pyrimidines cut. And uracil is in here, but it's not in DNA, and it's only in RNA. So if you remember pyrimidines cut, then you'll remember what nucleotides are a pyrimidine. And so there are rules about what can be paired with what. So purines are paired with pyrimidines. And so, but not just any one. So this is called Chargaff's rules. So let me erase this. Chargaff's rules. And all that states is that essentially A pairs with T down here, adenine pairs with thymine, and guanine pairs with cytosine. So right there. And one thing I forgot to mention, purines are a double ring. They have a double ring structure, while pyrimidines are a single ring. And so there's a couple criteria for genetic materials. And so let's talk about that. Let me clear this. So DNA it has to carry information that is necessary for the construction of an organism. The information / genetic information has to be transmitted from generation to generation, and it has to be able to be replicated, and finally it has to be capable of variation / mutation. So DNA codes for all of your features. The order of the nucleotides essentially codes for all of your features. And so let's talk a little more about the structure of DNA. And so this DNA model was created by Watson and Crick. And what they did was to discover the double helix model was they used an X-ray crystallography from Rosalind Franklin. And so as you can see right here, the DNA structure looks like a twisted ladder, and that's called the double helix. And so what's located on the outside in blue right there is sugar and phosphate. And so just to remind you, a DNA, a nucleotide consists of a nitrogenous base, which is adenine, thymine, a 5-carbon sugar, and a phosphate group. So 5-carbon sugar there. Sorry, it's actually not drawn like that. And it actually kind of looks like that, where this is the phosphate group, this is the sugar, and this is the nitrogenous base right there. And so right here, that's what makes up the backbone, so the part in blue. And the part in the middle, you see with the different colors, is the base pair. So that means that the base pair slash nucleotides is the middle right there. And so the orientation of DNA, its strands of DNA are anti-parallel. Which means that they have opposite orientations. So one side is oriented in the opposite direction of the other side. And one thing I forgot to mention, these base pairs are connected by hydrogen bonds. While the backbone, the phosphate groups, are held together by phosphodiester bonds.